There we go. Okay, so I'm going to talk to you very quickly about the genetics of peripartum cardiomyopathy, which we've learned quite a bit about in the last couple of years. So just as a primer and a reminder about peripartum cardiomyopathy, as I think you all know, it's a pretty significant disease, systolic heart failure with an ejection fraction less than 45, happening in the last month of pregnancy or the first few months post-pregnancy, usually in the first few months post-pregnancy, and the cause is unknown, although we've learned quite a bit about it in the last few years, so I think that goes in quotation marks nowadays. It may be rising in incidence in the United States, or we may just be diagnosing it more, but it's roughly 1 in 1,000, and that's true in the US, and it's true in most places in the world as well. And it's a significant disease, and it's because we're getting better at treating hemorrhage, treating hypertension, treating infection around the time of delivery, but not really so much at treating PPCM. The relative contribution of PPCM to peripartum mortality, maternal peripartum mortality, has actually gone up, and in many places in the country, it's actually now the #1 cause of maternal mortality around delivery. And we showed in a recent study actually that it accounts for much more than half of all cardiogenic shocks presenting around the time of delivery. So basically, if you see a woman in cardiogenic shock, chances are she's got peripartum cardiomyopathy. It's a bad disease, and it's also very strongly, just to follow up on what you just heard from Anant, it's very strongly associated with preeclampsia. This is a meta-analysis that we did a number of years ago showing that really across the board, it's at least 20% of patients with PPCM have co-diagnosed preeclampsia. This is probably more in real life because many of these were probably underreported. And actually this association and a lot of data that I don't have time to talk about has led us and others to propose that one of the mechanisms of how peripartum cardiomyopathy comes about is really through a vascular insult, much similar to the one that you just heard for preeclampsia, and hence the overlap with preeclampsia, by factors just like you just heard, soluble FLT coming from the placenta, also other endocrine factors coming from the pituitary, like prolactin, and causing a vascular insult to the heart that the heart cannot handle very well. So this is actually not what I want to talk about, but I want to put that in just background as to how we think about this disease. And thinking about this disease has led us to look at the genetics of the disease. So why did we consider the genetics of this disease? There are a lot of reasons. One of them is it's 1 in 1,000 people. What is different about that one woman from the other 999? That she develops peripartum cardiomyopathy and the other ones do not. And that frequently leads one to think about genetics, obviously. It's also dramatically more prevalent and in women of African descent, and with a worse outcome in women of African descent, and suggesting that, and of course it could be socioeconomic differences, but it suggests that there's something genetically different about the African background from the Caucasian background that makes this disease worse, so suggesting that there's a genetic component to this. There are a couple of reports in the literature of rare pedigrees where the disease runs in the family, again suggesting genetics. And perhaps most strikingly, there is a very strong, obvious phenotypic overlap with idiopathic dilated cardiomyopathy, which is basically the same thing as PPCM, but just showing up later in life and not triggered by pregnancy. And when you compare the two, the dilated cardiomyopathy is very similar, and we know that the idiopathic dilated cardiomyopathy has a very strong genetic component. So we decided to ask this question a number of years ago: could there be a genetic component to peripartum cardiomyopathy? And the way we did that is we were lucky enough to get samples from around the world, including from Dennis McNamara's IPAC study that you're going to hear about in the next talk. And we collated all these samples, totaling 172 from various different countries. 40% of them were African American, and that'll become relevant in a second. And we looked in these 172 patients for signatures of genetic modifications that are known to cause idiopathic dilated cardiomyopathy. So we didn't sequence the whole genome, we didn't even sequence the whole exome. We just sequenced 43 genes that have been known in the past to be seen as mutated in some cases of idiopathic dilated cardiomyopathy. So we definitely looked under the lamppost, and that's relevant and also important because statistically it allowed us to come to conclusions that we might not have been otherwise. So we take these genes, and basically what we do is this is for the people who are interested in this, you take all of the DNA, you cut it into little pieces, and then you bind it to a filter that has those 43 genes on it. And so only the red things stick. And that's what you sequence. So you don't have to sequence the entire genome. You just sequence the red parts that stick to the filter. And then we look for mutations, and we didn't look for any changes because there are lots and lots of changes in every single one of us. So every single person here would have at least one mutation in one of these 43 genes that nobody else in the world has, because that's the rate at which we generate mutations every time we have children. So that becomes a statistical nightmare. So we looked for mutations that we knew are likely to cause problems, meaning mutations that cause stop codons, truncating variants, mutations that cause stop codons or splicing variants or frame shifts that would lead to a truncated protein. So we're really looking at a very small subset of all the possible damaging mutations, but they're the ones we're sure about. And we found lots. We found lots. This is the list. I don't mean for you to look through every single one of these, but this is far more than we would have normally anticipated. And the vast majority of them are in this gene called titin. So what is titin? Most of you probably know what titin is. For the Europeans in the audience, it's not Tintin, it's Titan. Titan was named after these Greek massive gods, larger than life, that came sort of before the Jupiter ones that we all know about. And the reason they were called that is because it's a big protein. It's the biggest protein in the body. It's about 2 microns in length. It's as long as a bacterium, and it spans the entire sarcomere from the M to the Z line. And it basically has likely lots of functions, but one of the functions is probably just hold the sarcomere together. So you can imagine why a truncating variant, a truncating mutation in one of these proteins might actually affect the way the heart contracts. So that was striking. And then the other thing that was striking is that this is exactly the gene that was found to be mutated with relatively the same amount of percentage in large cohorts of idiopathic dilated cardiomyopathy. So we actually found the same thing as in cohorts of idiopathic dilated cardiomyopathy. And this was discovered a few years ago by the Seidman group, with whom we've actually been collaborating on this. So here's just a couple of numbers. And let's see, do I have a, yeah. If you just look at the numbers of total truncations we saw in our group, it was 30, and we compared it as a reference group. This is 60,000 publicly available whole exome sequences. And when you compare it to those 60,000, there's a number of truncating variants in those genes as well, but the percentage is much, much smaller, and the p-value is very strong. And if you focus in on the titans, it's even stronger, p-value 10 to the minus 10. There is 1% in the population, I'll come back to that in a second, but the 10% in our group is much, much, much higher. So on the other hand, if you compare it to a cohort of patients who have dilated cardiomyopathy, then it looks very similar. 12% of them have truncating variants in titin, and 10% of PPCMs have truncating variants in titin. So what I'm basically saying is that genetically speaking, PPCM actually looks a lot like idiopathic dilated cardiomyopathy. Now, why it is that some women would develop peripartum cardiomyopathy with pregnancy and others don't, and then develop idiopathic dilated cardiomyopathy later, is not clear at all. But genetically, they look like they're very similar to each other. That 1% is interesting. There's actually 1% of the people walking around with mutations in this gene, and they don't all have dilated cardiomyopathy, and that's a very interesting thing, and I have a minute, so I'll tell you about it. It turns out that the mutations are spread out differently. So here's the gene. It's big. It spans over the A band or the I band of the sarcomere. And it turns out that most disease-causing mutations are over here on the right side, whereas the healthy volunteers are mostly over on the left side. And that's because there's the exons, there's like 250 exons in this gene, and the exons in this part of the gene gets frequently jumped over. So if you get jumped over, you can imagine if you have a stop codon in an exon that you get jumped over, it doesn't matter. So you can handle variants here, but not so much here. And that's probably one explanation why this is the case, and it tells you why especially these large genes have to be interpreted with some subtlety. So in the last few seconds, just kind of mining a little bit deeper into some of this data, there's lots of interesting questions, and this is all post hoc analysis, so it has to be taken with a grain of salt. But you might predict that the women with a titin truncating variant would have a different prognosis than the ones without. And at least in a post hoc analysis, it sort of looks that way. We looked just in the IPAC study because that's the one where we had good clinical information. And when we look in just that clinical subset, Even though at enrollment, the EFs were more or less similar, at 1 year, the ejection fraction was much improved when you did not have a truncating variant compared to titrating with a truncating variant. So they seem to do worse. But again, it's post-hoc, so got to take that with a grain of salt. And then the preeclampsia question is interesting. Is there a difference between the two? And it turns out, again, post-hoc, but in this group, it turns out that every single case, almost every case of preeclampsia occurred in non-titin bearing variants, almost as if the two separated. So is it possible that there's two diseases here? One is preeclampsia associated and one is titin variant associated. We don't know yet. Again, it's post-hoc, but it looks like that might be the case. So just to finish up, there's a big genetic component here. We don't know how big it is because we looked under the line post, so it could be more than 10-15%. It looks a lot like dilated cardiomyopathy. How much does PPCM equal DCM is still not completely clear, but there's a big overlap here. And maybe it correlates with poor outcome and the absence of preeclampsia. So I will stop there and maybe take a question or maybe later.